All right, thank you all for coming. I was very surprised when I was standing in here getting ready to talk, and I believe my entire extended California family is here tonight. That was very nice. I wasn't expecting that, and seeing so many familiar faces out there, it's really nice that you all came to this talk. My goal is to entertain you. I wrote this talk to entertain myself because, as Jeff said, I was a little reluctant to do this, but I will make it fun for myself, and hopefully you are entertained as well. Before I get started, I have a couple thank yous, because when you work for a place for 35 years, there's a lot of people that make it possible and help you along the way. So first, I'd like to thank you, Jeff. You provided an amazing amount of funding over the years so that I could do on the lake what people do on vacation. I went out every day. I had 2 boats at my disposal, and I did what I loved. That was great. And by providing that resource, what you were really doing was giving me the opportunity to explore the scientific things that I found interesting. And when the team is out there on the lake, we're looking at the lake ecology all the time. And if we notice something, we had the freedom to actually explore that. And it turned into several different projects. And if Jeff had been more of a micromanager and said, no, I just want you to get the job done, don't burn any fuel that you aren't doing for work, that opportunity wouldn't have happened. So thank you, Jeff. Appreciate that. Another person I need to thank is Bob Richards. Did Bob make it here tonight? Is he around? Well, Bob was for anybody who doesn't know, Bob Richards was the captain before I was, and I overlapped with Bob for about 15 years. And what Bob taught me was, first of all, he was my mentor for doing lake work, for doing limnology out on Lake Tahoe. And it was everything from using Van Dorn samplers or using 13 Van Dorn samplers all on a single line. All of the different equipment that we had, and a lot of it back then was analog equipment. We didn't have digital samplers. But it was more than teaching me how to use the equipment. Bob had that deep understanding of the Tahoe knowledge. And so what he was teaching me really was how all of that data that we collected every day out there fit into the bigger picture of what we're trying to do at Tahoe, what the overall goal of the research was. And so I really appreciated that, because I gained an enormous amount of insight into why we take all these samples and what we do with this data. And Bob was incredibly diligent. So he passed that on. When you lower an instrument down to the bottom of the lake, 450 meters deep, and it doesn't collect data, Bob would do it again. And then he got home 45 minutes later. But the integrity of that data set meant a lot. So he passed that on to me. And hopefully I was able to kind of fill those same shoes to a degree, where Tahoe has one of the longest continuous limnological data sets in the world. And that really was a tribute to Bob, and then Bob's insistence that I also repeat something down to the bottom of the lake if I had to. The next person I'd like to thank is John Reuter, who can't be here tonight. But John was my academic mentor, if you will. And he taught me how to look at these large data sets that we collect on a project and really tease out what the important parts were, and then put them into reports so that you can pass them on to the funding agencies in a digestible way and find the really meaningful parts of the data sets. John was very, very good at this. I think people actually read his reports, which is amazing. So one example of how John did this, he always did it with fun and with a sense of humor. And he and I had just finished the MTBE fuel additive 2-stroke engine ban. We were looking at all the fuel that comes out of 2-stroke engines and the emissions that ended up in the lake and this fuel additive. And so I was down in his office in Davis, and we spent all day crunching numbers. And he After crunching all the numbers for the lake, we had the lake concentration for the surface waters. What is the amount of MTBE in the lake? And then we took all of the data for the hours of two-stroke engines that had been done during that year. And if we assume certain percentages, they should come out somewhat close, that the MTBE was related to the two-stroke engines. And shockingly, it actually did. We had these two different data sets and it matched up really nicely. So we were shocked by that. But that wasn't quite good enough for John, because we're looking at a few parts per billion of MTBE out in the water. He said, you know, the people that do water quality monitoring stuff, they get it, but the public doesn't. And some of the agency people just don't get what that concentration means. So we have to do one more calculation. I was like, oh God, here we go. And then Bob Bridger's on my shoulder saying, diligence, come on, we do these things. So John said, well, there's the other fuel additive that the states around California use that we don't, and that's corn ethanol. So let's take the concentration of MTBE and assume that it was ethanol. And we're going to calculate how many gallons of Tahoe water you would have to drink to become drunk. And I don't remember what the number was, but it was something like 10,000 gallons in an hour. But that was the way John thought about data sets and really translating. So it was quite funny. And before I get into my talk, there's one more person I have to thank. You always save the best for last. So I really want to thank Denise, my wife. My understanding is that TERC is trying to hire someone to replace my position. Denise knows more about my position than anybody else out there. So she spent many, many nights over the dinner table listening to scientific geekiness about different species and how they interact in the lake and what zooplankton is doing what with what algae. And she listened to it all very patiently. And she also put up with, as Jeff was describing, the weekends, the long nights, sometimes the day, night work where I'd show up 4 days later. There were also times when I had some extensive travel. So for 7 years, I spent 6 weeks a year in Mongolia. And you can't do that in a career unless somebody is holding down the home fort, especially when you have a family. So that was her contribution to TERC, and I want to make sure that was recognized. Can I get rid of it? There we go. Okay, so you see it says 37 years of thinking outside the box. I've been in Tahoe for 35, but I did actually work in the field as a researcher for 2 years before I got here. So to make a bigger number, I just went ahead and put 37. If anybody here, the original flyer that went out for this talk was about the interaction between the decline in Mysis shrimp population and the resurgence of cladocerans and the clarity of the lake changing. If anybody is here to hear that talk, there's an exit over here and over here. I won't be offended, but that's not what I'm going to talk about tonight. That story will emerge over time. We've told half of it, the other half will be exciting to hear, and it's probably going to be about a year out. Think outside the box. There we go. Okay, so when I first started working at Tahoe, the only office we had was in the old fish hatchery building, and it was rather primitive. Even by standards back then, it was a bit primitive. So we had one computer, is about the size of a suitcase. We had a dot matrix printer. We had one telephone that was corded to the wall, and we would hand it back and forth between desks. So Scott, Bob Richards, our chemist, Mark Palmer, and I would, if a phone rang, whoever's desk it was on would answer it, and then whoever it was for, we just pass it along. We knew a lot about each other. We also had, communication back then, obviously we didn't have cell phones. So the communication back and forth of the boat was the marine radio. And the marine radio was on channel 16, 24/7. And so when you're working at your desk in the wintertime, it was pretty quiet. There wasn't much happening on the lake. But in the summertime, there was just constant chatter. And it would be fishing guides talking to other fishing guides. It would be the Coast Guard going out on a mission or a rescue, or talking to boaters, or it would be tourist boaters trying to figure out where they rented their boat and get back to the marina. So while it was entertaining, it was just this background noise that was always going on. And you just got to the point where you didn't pay that much attention to it. But it was kind of like when you're at the park and your kid starts crying. And all the other kids have been crying all day long, but suddenly you hear that kid crying and you know it's yours. So if I were to say right now, this is WHX 954 calling WYK 3721. Scott just got up to answer the phone. If Bob was here, he would have also. Those were the call signs of the LeConte calling into the base station. And it might be for, hey, I'm back off the lake now, everything's safe. Or it'd be, I have samples, or I forgot to bring an instrument down. Can you bring it down to the marina? Or I'm out in the middle of the lake and the boat broke down. Can you grab the other one and come get me? So really, throughout my career, it's going on still. We had this enormous technological boom that happened. It happened very, very fast. So what was going on is that all of this instrumentation was happening, and it really changed the way that ecological research was being done. So we had things develop like environmental DNA, where you take a dip sample of the water, run it through an instrument, and it'll tell you what species are actually in the water. Doesn't matter if it's a lake or a river. I read an article recently where they're actually starting to do this in Africa with air samples. And they run the air sample through a DNA analysis, and they can tell what species are upwind when that was taken. So these kind of technologies were taking off. And when you look at our primitive office compared to this, we now have laptop computers out on the boat. We're running robots through the lake that go out and fly around for a month, come up at night, download their data through satellites. We have ADCPs, acoustic Doppler current profilers. They're in the lake and they break up the water column into a bunch of different segments and tell you what direction and at what speed the water is moving in each of those different segments. We have instrumentation in the lake that is reporting the temperature, the chlorophyll, the dissolved oxygen, multiples around the lake that are reporting data every second. So it's a massive change in the way data was collected. And the only problem with that, for me, was that it didn't apply directly to ecological research from the way that I was trained as using direct observation. So it changed the way papers were published. And I submit the idea that Jane Goodall, who spent thousands of hours observing chimpanzee behavior in the field, would have a difficult time publishing her papers today if she didn't have the DNA analysis to support all of the observations that she made with family dynamics in chimpanzees. And similarly at Lake Tahoe, where we rely on these papers that were written in the 70s about the change in the aquatic ecosystem. Those papers were all written on direct observation. But it would be hard to do those now if we didn't have the stable isotope analysis that's done in the laboratory, looking at the carbon and nitrogen signals to tell us where actually these things are deriving their energy. We can go out and observe it, but there became this reliance on this digital black box data. And it kind of pushed against this direct observation that I had done for so long. So I'm going to tell 3 stories tonight. If anybody's offended, feel free. The exits are again over here and over here. It probably isn't going to make you any more comfortable, but all 3 of these stories are based on direct observation. No black boxes involved. So the first one is a menage a trois under the Tahoe moon. And thank you, Raph, for making this photo available. Raph was driving the boat, and it was at the end of a long day. And I don't think he really wanted to do another 30 minutes so I could get a photo of the moon over the LeConte, but I appreciate it. So this will come up in a minute, but this photo was taken in the fall. So when I first got to Tahoe, this was one of the first projects I worked on. And my job was to really reconstruct or figure out the fisheries part of the Tahoe food web. Who's eating whom? What areas of the lake do they use? Why aren't all the different fish eating each other? What are the gaps that are there? And to do this, I had to collect a lot of fish samples. So the easiest way to do it was to go out on the guide boats, and they were already collecting the fish, and I could just clean the fish that they had collected. It's a very efficient process. And I really have to give a tribute to Mickey Daniels, the Big Mac charters. He supported all Tahoe research, and he really supported me throughout my career. He was always calling me if there was some anomaly that he saw out there or something interesting, but he let me clean fish on the back of his boat. And sounds like a bonus I was giving him, but it really took longer. And these people had been out since 4 in the morning catching fish. And then the researcher shows up and he says, well, it's just going to take me, another 45 minutes to clean all your fish for you because I have to do it scientifically. So I would take all the stomachs and I'd measure the fish and I'd put the stomach in the appropriate bag and take it back to the lab and find out what they'd been eating. And when I got through with this, 1,000 fish or more that I'd been looked at, I had this giant data set. I said, okay, now what's important? So John Reuter's training came in and I'm like, okay, I got to figure out what's the important part of this data set? What am I going to talk about? And there was the basics, but I saw this anomaly that was occurring. And I'm going to give you a little bit of background so that it makes sense. I think most of you probably know this. This is kind of the classic Tahoe introductory food web situation. So we have deep living lake trout. They're down about 60 plus meters in the lake. They're bottom oriented. And then we have the shallow living kokanee salmon that are in the top 100 feet of the water. And they're pelagic, meaning that they're kind of out in the blue water. They're not associated with the shoreline. So the lake trout and the kokanee don't interact. And between those two are the Mysis shrimp. These were introduced in the 1960s. And the Mysis shrimp don't like light. They stay away from light. So during the daytime, they're down on the bottom with the lake trout. And the lake trout get to eat them a bit. And then when it gets really dark at night, they make a migration up to the surface. And if it's really dark, they go all the way up into the surface waters and they feed there. Unfortunately, the kokanee salmon are sight feeders, and the shrimp don't get there until the kokanee have gone to sleep. But the shrimp are really good at eating their zooplankton favorite food, Daphnia. So that's kind of the background story. And then the sunrise starts, the shrimp go, ooh, it's getting dangerous because the light's out. So they head down to the bottom. And the kokanee salmon wake up and all their food is gone, and the shrimp are gone as well. So I did some work with Dave Beauchamp. He's at Washington State University now. This was done back in the 1980s. And it's another graphic that shows exactly that situation. So the Mysis shrimp were put in 1965. They didn't really get established lake-wide until about 1970. And that's when we lost the Daphnia, the food they were eating. And the kokanee salmon, because that was their favorite food, their size went from 15 to 20 inches down to about 10 to 13 inches per fish. And it was because this loss of their primary prey. So that just supports its actual true data that supports my cartoon slide just a second ago. But this was the rule and the understanding of the fish ecosystem when I got here. And rules are not fixed. They're meant to be broken. So I found when I was looking through all these fish stomachs that lake trout sometimes actually do eat kokanee. And kokanee sometimes actually do eat Mysis shrimp. And when I looked at my samples, it was very rare that it happened, but it happened a couple of days a month and only in the winter. So the kokanee, when I would clean a lake trout stomach and it had a kokanee in it, the kokanee was really freshly eaten. It still had scales on it, hadn't been digested at all. So being the geeky scientist I am, I went ahead and cut open the kokanee, and 100% of the time, they had Mysis shrimp in their stomachs. So this whole understanding of this fish food web that wasn't supposed to be happening was happening a few days a month in the wintertime. And you can think of it kind of as like the nesting Russian dolls. The kokanee has the shrimp in it, and the lake trout has the kokanee in it. So because we didn't have the internet, I went to the paper internet, which was the Farmer's Almanac. And I looked up these winter dates for when this had happened. And it turns out it happens in the 1st to 4th day after the winter full moon. And indeed, the moon creates the Tahoe fish menage a trois. So the reason this happens is that in the wintertime, after the full moon, you get complete darkness in the Tahoe Basin. And then the almost full moon pops over the Mount Rose, and it's like the lights came on for the kokanee salmon in the top 100 feet of water, and all the shrimp are there. And so the kokanee start feeding on the shrimp. Well, as soon as that moon pops up, the shrimp are like, we got to get out of here. This is dangerous. So they start heading down to depth. Well, the kokanee see all this great food resource, so they start following them down. The lake trout are down there, and all of a sudden it's raining adult kokanee and Mysis shrimp. And so they're opportunistic feeders, they start gobbling up all the kokanee. So that was really the discovery that came out of it. And the reason it doesn't happen in the summertime is that there's enough lingering twilight in those days after the full moon that you don't get dark enough, the basin doesn't get dark enough, so the shrimp don't come all the way up in that zone where the kokanee are. And when you get far enough in toward the new moon, where it does get dark enough, when the moon pops over Mount Rose, it doesn't provide enough light for the kokanee to then feed on the Mysis. So it's a winter phenomenon. It's a three-way food web based on a lunar cycle. When I started looking into that, I started researching papers that had predator-prey relationships with the lunar cycles. And there's a lot that were based on two species where a predator-prey interaction changes or somehow moves around with the full moon. But I couldn't find one that dealt with three species and a lunar effect. So that was pretty exciting to me. Again, geeky science, but it kept me going and loved what I was doing out there. All right, moving on. Proof that size really does matter. Nobody's left yet, so I think I'm okay. So I had a project on Fallen Leaf Lake, and it was a cooperative project with University of Nevada, Reno. I was working a lot with Sudeep Chandra on this project. And it was funded by the US Fish and Wildlife Service because they were getting ready to reintroduce cutthroat trout into Fallen Leaf Lake. And their goal was ultimately to introduce cutthroats into Lake Tahoe as well. But they wanted to start with a smaller lake. It's much easier to study. And so we were hired to look at that. We knew a fair amount about what some of the issues were going to be. And we knew lake trout predation was going to be a really big deal. because of the work that was done at Yellowstone Lake. In Yellowstone, lake trout were introduced. They had a native cutthroat population, and the lake trout were just devouring the cutthroat. So they were harvesting lake trout, trying to maintain their native cutthroat population. The project we were working on is a little different. We had no cutthroat, lots of lake trout, and we were trying to figure out how you would reintroduce them. But the similar situation, we knew predation was going to be a big deal, largely because lake trout can eat food up to half their own body length. So the first thing we did is we looked at what is the lake trout diet in Fallen Leaf Lake before the cutthroat go in. And it was kind of what you'd expect. I mean, they eat some Mysis shrimp, they eat some fish, they eat some crayfish. But if you look at the vertical graph, that's 1 gram of food per lake trout. Very little food. So we knew that the lake trout population in Fallen Leaf Lake was stunted by food resources. They weren't growing as fast as they could if they had more food. That previous graph was fish less than 20 inches, lake trout less than 20 inches. When we look at the whole pool of lake trout that were out there, so this includes some of the large fish too, they definitely consumed a lot of fish, but the fish they were consuming was their own species, so they were cannibalistic. And the lake trout really, they don't care what it is, they're opportunistic feeders, so they were feeding on smaller lake trout. And that was really just the biggest fish in Fallen Leaf Lake. So when we put together the whole spectrum of what does the fish size look like throughout, what does the lake trout size look like throughout Fallen Leaf Lake, the average lake trout size is only 15 inches. And more importantly, only 3% of the lake trout grew bigger than 20 inches. So that had a lot of information for us when we started looking at how you would start managing this reintroduction of cutthroat. The black line is the growth rate of lake trout in Lake Tahoe, and the blue line is Fallen Leaf Lake. And what I want to point out here is that here's 20 inches. It takes about 5.5 years for a lake trout in Lake Tahoe to get to 20 inches. It takes twice as long, it takes 11 years for one in Fallen Leaf Lake to get to that size. So if the lake trout were food limited in Fallen Leaf Lake, what we didn't want to do was drive that blue line up to that black line. So the first year, the Fish and Wildlife Service put in 36,000 pounds, which sounds like a lot, but on stocking measures, that's really not huge. They put in 36,000 pounds of cutthroat trout. And these cutthroat range from about 5 inches to about 9 inches. And they definitely contributed to the lake trout diet. In fact, the lake trout diet went up 800% by mass. So we knew this was unsustainable. If you continued to do that, you would just grow your lake trout bigger and bigger and bigger. So before we made our final conclusions, we went and looked at what would be our bycatch. So when we were setting the deep water gill nets, what other fish showed up where the lake trout are, down in the deep part of the lake? And it wasn't, the revealing thing wasn't the species that they were eating down there. But if you notice, all the fish we caught down in lake trout depth zone are over 10 inches long. So what it told us again was, if you are 10 inches long, you can survive in Fallen Leaf Lake with the current lake trout population. If you're smaller than that, you better find another lake to live in. So it turns out that if you were 10 inches long as any fish, you would escaped 97% of your predation risk. So our recommendation to the Fish and Wildlife Service was, you need to grow these fish in the hatchery to 10 inches before you put them in Fallen Leaf Lake, because you don't want that blue line becoming the Tahoe black line. Fallen Leaf Lake is a very small community, and it's hard to get in there as tourists. So we knew that when we were going to be gill netting in there, we were going to be very obvious. So rather than try and hide, we wore flamboyant Hawaiian shirts. We made it known we were there. And these two fishing guides came up to us, and they weren't super happy we were gill netting because they spent 30 years catching lake trout. And they did it the exact same way for 30 years. The beauty of it was that they wrote down the length of every fish they caught over 30 years, whether they kept it or not, and they released a lot of fish. So fortunately, we were able to make friends with them, and they gave us all of their fishing records. We entered it in the computer, and they were complaining that the lake trout used to be bigger, and they've shrunk over the time that these guys have been fishing. You hear that from fishermen in just about every lake. But these guys were actually right. So this is their fishing data. And in the 70s and before, they were catching fish that averaged around 18 inches. And starting in 1980, the fish size they were catching dropped dramatically until it was what we were seeing in Fallen Leaf Lake with 15-inch fish. So we weren't quite sure what was going on with that. So we got all of the California Fish and Game stocking records for Fallen Leaf Lake going back to 1950. And California Fish and Game had a policy change in 1980 where they switched from a rainbow trout management program to a kokanee salmon management program. They managed the lake differently. And what that did was when you manage it for rainbows, you put in catchable size rainbow trout. These range from about 5 to 9 inches, exactly what the Fish and Wildlife Service had put in with the first batch of cutthroat trout. And Cal Fish and Game was putting in anywhere from 30 to 60,000 pounds of rainbow trout. They were artificially raising the length of lake trout by feeding them. And that's what we see up here. When you do a kokanee management program, you strip the eggs out of the salmon, you take them to the hatchery, you fertilize them, and as soon as those kokanee are fry size, so they're about 1 inch to 2 inches long, that's when they get re-released back into the lake. And the total mass of fish you're putting in is obviously much smaller. So what happens is those little teeny fish that go into the lake, they don't fortify the lake trout. They're divided among the whole lake trout population. And there's just a small, perhaps 2 to 3 gram fish, as opposed to these bigger fish that would actually grow your lake trout bigger. So this told us a lot, because not only should Fish and Wildlife Service put in 10-inch fish, because size does matter, and bigger is better, they should also stock little fingerlings. Because while they might lose a lot of those fish to the lake trout population, they're not going to grow the smaller lake trout up so that they're like the Tahoe fish. This is just what happens. You put in small cutthroat, you grow your lake trout. You put in bigger cutthroat, you grow your lake trout. You have to put in bigger cutthroat, and you just get behind this spiral if you didn't follow this management plan, where you'd never be able to get ahead of it. And you'd probably have to wait a decade if you didn't do it the right way to then go back and start over again. So they planted 10-inch fish. And it was really interesting because the lake trout that were less than a foot long, no fish ended up in their diet. Those that were 12 to 14 inches, they ate cutthroat trout, but they were only eating those little juvenile cutthroat trout. Even the fish less than 20 inches were only eating those little juvenile cutthroat trout, and it was 5% of their diet. The big fish, the ones over 20 inches, definitely got a bonus. They were eating some of those 10-inch cutthroat, but we're talking about 3% of the population that were big to begin with. So we're only growing the big guys bigger, and we're preserving that growth curve for the lake trout. So that story worked out really well. The real take-home message was that if you understand the system and what the players are in it, you can make a management strategy that could work even in a predator-dominated system, as long as you remember the size matters. I had one person leave, but it was a director's wife, so I think I'm okay. So the next story is how a little sewage can ruin date night. Back in the early days of Tahoe work, it was the Tahoe Research Group, and Charles Goldman was running the group. And occasionally we would get requests for researchers to do really small projects. And it was the type of thing, they were like $3,000 to maybe $6,000 projects. And if you ran those through the university, they're going to take 50% overhead. So your 3,000 became 1,500, your 6,000 became 3,000, and you can't even do the project. So a lot of those really small ones would go through a consulting firm and get handed out to postdocs or occasionally staff. So I was fortunate that John Reuter decided I was capable of doing a fish study down in San Bernardino. Well, it was in a sewer outfall. So thank you, John. This San Bernardino watershed, it's the Santa Ana River flowing through San Bernardino, and it is a desert dry wash. So it's really sandy soils, it's flash flooding type situation, and that was only exacerbated over years as they paved 90% of San Bernardino. So when you had a thunderstorm in the mountains, the flow going into the Santa Ana River was just huge. And in the summertime, or most of the year when you didn't have a thunderstorm, or even the snow runoff, that water was sucked out for industrial use and domestic use in San Bernardino. So the Santa Ana River had very little flow. So this sewer treatment plant was pretty innovative down there. It's called a rapid infiltration and extraction plant. And what they did is they collected the wastewater, they put it in this couple of different large ponds, and they used the natural percolation effect of that sandy soil where it would go down 300 feet filtering through the sand. Then they'd rapidly extract it, run it through a UV filter, and dump it back into the Santa Ana River. And 80% of the flow was treated wastewater going into the Santa Ana River. So the problem was it's really nutrient-rich water, and they would get big algal growth on their UV lights, their UV light array. So occasionally they just have to shut off their outflow. Well, that turned off 80% of the river until they cleaned their light arrays. Then they could turn the water back on. And the Fish and Wildlife Service didn't really like this because the threatened Santa Ana sucker is sitting in the river and suddenly 80% of it goes away. So they were really worried about habitat loss. And we're talking a matter of minutes where the river would go. As you can see, the blue line is full flow. And when they shut off the treatment plant, it went to what the pink line is. So right below the outfall, the river channel basically just went dry. Where you had the other stations, I just picked 3 of them that we monitored, where you had the other 20% of the flow, you still had water in the river, but there was a dramatic decrease in the river channel. And just for people that get into this kind of thing, the vertical scale is millimeters. So the river went from kind of a max depth of 400 millimeters to 150 millimeters. We're talking 15 centimeters. So we're not talking about a deep, wide channel or something. But when we surveyed we found the Santa Ana suckers in the deepest pools in the river. And so our job was, they're going to shut off 80% of the river flow, and we're going to walk down the river, and we had buckets of water. Any Santa Ana sucker we find, we're going to pick it up and put it in the bucket because it's a threatened species. And then when the river turns back on, we'll release them back into a pool or wherever we found them. So we walked down the river, and there were a lot of stranded fish, but they were all largemouth bass, tilapia, bullhead catfish, and mosquito fish. We could not find a Santa Ana sucker. We saw them in the pools, but we could not find a stranded sucker. So it occurred to me when we're doing this that all of the fish that were stranded are predators on Santa Ana suckers. Bass for sure, even tilapia will eat small fish, and certainly catfish will. So my conclusion for this part of it was that these shutoffs actually were somewhat beneficial to the Santa Ana sucker because it was taking out all the invasive non-natives that were predators on them. So why weren't we finding any Santa Ana suckers? The Santa Ana suckers evolved in a flash flood zone. So they use the pools as residents, and we would see flows. I just pulled this off of a more recent graphic, but it shows it somewhat. When I was there, the flows would go from less than 100 CFS to 5,000 CFS and back to under 200 in 24 hours. So that's the flow regime, the dynamics of the Santa Ana River. And the Santa Ana suckers, they'd been living in there for thousands of years. They knew what to do. So they lived in the pools. The bass were not quite as brilliant. There was another problem that Santa Ana suckers could face. So I threw this in a little bit in my report to the treatment facility, and they were very happy to hear that their shutdowns were actually potentially beneficial. But we were doing the in-stream flow measurements, and we had our tape measure stretched across the stream. We're standing in the stream, we have our flow meters there, and we hear this noise, and this Jeep was coming up the river about 20 miles an hour. And apparently it wasn't a one-time thing. It was common for these off-roaders to go racing up the middle of the Santa Ana River. So I threw in the report that recreational opportunities might also be detrimental to these Santa Ana suckers. But yeah, the other part of the study was looking at the spawning. So again, thank you, John Reuter. I got to snorkel in the sewer outfall of San Bernardino. And when I got to this pool, this was probably the deepest pool. It was scoured from the releases that they did. And in the spring, I was there at spawning time, or I thought I was. I snorkeled this pool, and I'm expecting to see suckers spawning there. And when suckers are spawning, they get this really dark stripe on their side. In Tahoe, it's a red stripe. Santa Ana's are a black stripe. They get little bumps called tubercles on their fins, and they act like they're spawning. They're together near the bottom. A bunch of males swimming around. When a female approaches the gravel, all the males dive in and try and fertilize the eggs. And I couldn't find any of that behavior or any of the suckers looking that way in this outfall. So at first I thought, well, I probably missed spawning season. I'm either too late or too early. But Santa Ana suckers, again, this amazing adaptation to living in this kind of environment, they spawn multiple times in a single year per summer. Really, it's over about a 2 to 3 month period. And they release thousands of eggs over that time. And the eggs, the fertilized eggs, hatch and develop into little teeny fish in 36 hours. So they don't have to rely on the eggs surviving in the gravel because there's these huge bedload shifts up and down the river that would just destroy the eggs. So once they're fish, they can seek out this pool habitat and these fringe habitats that allow them to survive in these crazy flow patterns. So again, lots of eggs, multiple spawning times, and super fast incubation times. So what I did when I decided I'd go upstream to where the 20% of the natural flow happens to see if I could find spawning there. And I snorkeled a pool there that was underneath a concrete apron underneath one of the major highways that ran through San Bernardino. And there, all the suckers were colored, they were spawning. It was clearly evident that there was spawning going on. So I thought, okay, I didn't miss it, this is great. So I walked downstream in the main channel and I found these like 3 millimeter long little teeny suckers, Santa Ana suckers. So not only were they spawning, but some of those eggs had already hatched and there were fish living in the river. So I started to question, well, I'm seeing 30, 40, 50 suckers in the outfall pool that aren't spawning. And what had occurred to me is that they had probably become sterilized based on the water that was being released from the sewer treatment plant. There was a lot of work being done at that time with environmental toxicology, and hormone replacement therapies were big. There's also chemicals that can mimic estrogen compounds. And so fish that were exposed to those, the males were actually growing ovaries on their testes and basically becoming sterile. So my theory was that because these suckers are very localized to their pool habitat so that they don't get flushed downstream to the ocean, they spend a lot of time in this water and they probably had become sterile. Even, especially when I found the same fish just 2 miles upstream actively spawning. So I ran this concept by the water treatment plant lawyer who is running the meeting just for the water treatment plant. And she said very clearly, you were not hired to look at that. You are not an expert on environmental toxicology. You did not run any samples through a laboratory, and that will not end up in your report. The message is very clear, and it didn't end up in my report, but now you all know about it. So that is how you can ruin date night. Unfortunately, I only had to snorkel in this pool for maybe 20 minutes, so I think I'm still good. All right, so all of those stories about those 3 different projects, it was all observational science. And we really didn't use any digital data to support those findings. So I guess kind of my message is that get out there and get your face wet, hike through your environment, study in the natural environment, look and observe what your species are doing if you're an ecologist. And then you'll have the understanding when you use that black box to question that data or put it in the right context and framework for what you've seen out in the field. So that does conclude my scientific portion of the talk, but there's definitely, I'm retiring. I have 4 more days of work. So I wanted to maybe head off a few questions I might get about, what was the best part about work? What was the worst part? That kind of stuff. So I put together just a couple of slides here, and there's a few things I'm not gonna miss. There are times when the boat breaks down. And the picture on the left is the LeConte getting towed back from Emerald Bay. Not too long ago, we were down in Emerald Bay doing night work. And we had started probably around 7:30 or 8. And we're just getting ready to wrap up. And the power takeoff for the hydraulics that's attached to the front of the engine blew up. And Brandon and I had a 75-pound instrument over the side of the boat, down about 150 feet. on a steel cable and no way to bring it in. So I used one of the tricks that Bob Richards had taught me, and we used some Jumars and some lines, and we got it all in, but the boat wasn't going anywhere. So we called the other boat back, and it came and got us, and we towed it back to the marina, and we all got in around 5:30 in the morning. Fortunately, we only had a little bit of work to do that day. Katie and I were headed out on the lake one day to do paraphyton sampling, and we got a call that someone had found a rather large buoy in their backyard and thought it might belong to us. These are not fun days either. We diverted what we were doing. We went and found the buoy, and the two of us somehow managed to get this thing back into the water, hooked up to the boat, and towed back to a safe place where we could then anchor it in shallow water. And then we got to make the call to NASA that they had just lost about $30,000 in equipment. The other picture is pretty self-explanatory. I'm not going to miss winter diving. That's what my feet normally look like after a winter dry suit dive. You just can't stay warm enough. So I won't miss that. Do we have volume on the will it come through if there's a video with volume? Okay. Everybody be quiet. I won't miss cold nights on the lake with wet gloves either. We'll try that again. So everything you touch out there in the wintertime, if you have a wet hand, it just flash freezes to the boat. I won't miss that. But there are things that a lot of things that I am going to miss. Way more I'm going to miss than I'm not. I'm going to miss playing with big fish. I got to travel all over the world, thanks to Zeb Hogan at University of Nevada, Reno. I tagged along on a bunch of his projects, and he was filming a TV show for Nat Geo. So the two of us would go ahead of that, figure out what the story was with the big fish. And then I would take off and he would get to do his TV show and tell the story. I caught one of these fish, but I got to play with them. That was really the fun part, because I was just a fish guy from the start and really liked it. So I will miss handling fish that are bigger than I am. I will miss doing secchi readings at Alibi Aleworks. I will miss doing interviews. This was probably the one that I said the most right things on ever. And I will just miss the joy of spending so many great days on one of the most beautiful lakes in the world with some really fantastic people. We've had a lot of fun out there. It doesn't matter if we're doing the research, we're doing safety training, whatever it is. It's just fun. We have a good time. We have a great crew, and it's been a wonderful time. So with that, thank you all who have participated. Thank you for coming. And thank you for making a really fantastic career. So I will take some questions if anybody has them. Excellent. Yes.
Arrived here, or were they introduced purposely, or?
They were introduced intentionally to feed the deep living lake trout and the shallow water fish. And it did work well for feeding lake trout. That's why that black line on that graph shows that lake trout and the smaller sizes of lake trout in Tahoe grow much faster than they do in Fallen Leaf Lake. That's because of the shrimp. Unfortunately, they don't feed the shallow living fish, which tend to be sight feeders, and the shrimp just aren't there when they can see. So it worked about 50% of what they'd hoped. Yeah. So the cutthroat trout are our native trout species. And so it was a reintroduction effort to get the native fish back into the system. And they realized that it was going to be really difficult with the angling public and the popularity of fishing in Tahoe and the surrounding lakes to eliminate the lake trout, to eliminate rainbow trout, which can hybridize with cutthroat. So they were looking for management strategies that would allow them to have a viable living cutthroat population without getting rid of the others. Yeah.
Are the lake trout what we call Mackinac?
Yes, lake trout and Mackinac are the same thing. Yeah. And back.
So the Institute Service is looking for a boat program manager in whale.
Thank you. Yeah.
Tell me about the young man drinking the Budweiser.
Yeah, so as kind of a requirement, if you're going to become a field biologist, I think, is to learn to do that at a young age. So I'll just give my dad credit that he got me started as a biologist early.
Somebody online wants to know, what is your favorite fish?
Oh, that's a tough one. The honest answer is my favorite fish is the one that I am studying at the time. So when I was looking into arapaima, these giant fish from the Amazon, they were absolutely my favorite fish. And then I went to Mongolia and studied taimen, and they were absolutely my favorite fish. So it shifted over time because as I learned about these different species, they're fascinating. They all have these really interesting characteristics that they've adapted to live in their environment. It's really fascinating. But as strange as it is, one of my favorite fish, just generally, is goldfish, carp. Yeah.
Are they currently putting Brant into Tahoe? Because I've seen, put fish at San Harbor, but I'm not sure.
Yeah, so that project was done. We did our portion of that project in 2002 through 2004. And the Fish and Wildlife Service wasn't real happy with our answers. So they hired somebody else to redo our project. And that guy had a lot of integrity, good researcher, and he came up with the exact same answers we did. So they moved on from there and they started actively harvesting lake trout out of Fallen Leaf Lake to give themselves better opportunities. And maybe against better judgment, but they do have timelines they have to deal with as federal agencies. They went ahead and started putting some cutthroat into Tahoe as well. But the size structure of the lake trout in Tahoe, to my knowledge, hasn't changed at all. So what they're probably doing is feeding lake trout. So if you know when they are stocking cutthroat trout, get a big cutthroat trout lure and go fish for lake trout. Yeah.
You talk about, and it's kind of off topic here, but the nice little, oh crap, I just forgot the little crawdads.
Oh.
Like, when were those introduced and why are they bad?
So they were introduced in 1923, and it was primarily for a food source for people, for the resorts. And they're fairly benign. They're scavengers on the bottom, and they kind of lock up nutrients and things away from the rest of the food web. So it's hard for me to come up with a really negative or positive thing about the crayfish. They're kind of there, and aside from locking up an enormous amount of calcium in their carapace and nutrients, they don't do much back to the lake that I'm aware of.
But are they still bad? Like, is there a name?
They're delicious. Yeah, I think scientifically, ecologically, they're probably not bad necessarily, They are introduced. Every introduced species has an impact. So in that sense, maybe ethically, yes, they're bad.
What are you going to do in retirement?
All right, I am going to spend more time exercising when I want to. I am going to paddle a kayak and maybe troll one line of monophona instead of pulling in 100 in a gill net. I don't care what that fish ate. I will eat it or I will release it. So yeah, fishing, kayaking, swimming, mountain biking, spending time with family that is here and family that isn't, and traveling. I have a trip, trekking trip to Nepal planned for October.
Are the Mysis shrimp still declining in the Tahoe, and any idea on these?
So the Mysis shrimp population declined, and it seems to be pretty stable at a very low level compared to what it was before. We think that the population starved to death. That's our best guess right now, but that research is still out there, and that's part of the story that'll evolve. The reason we think that might have happened is that all age classes of Mysis in Tahoe died at the same time. It wasn't like we lost our adults and the juveniles were still there, or we lost juveniles and adults were still around. We lost every year class, and Tahoe has a 4-year life cycle. So we anticipate that if they were able to rebound because they had food or whatever conditions took them out, if that went away, it would take about 4 years for them to come back. So I'm thinking their populations, based on what we know, maybe back up again in 2026, somewhere in that range. There's a question way in the back. I heard it, but I couldn't quite understand it. All right, I think I'll wrap it up there. Thank you all very much for coming.